Hi everybody, it's a pleasure to be here. I'm Carter Cliff with Papillon Therapeutics. Papillon is a clinically validated platform delivering transformative multisystemic medicines. And the high level on the company is that the company's lead program, or first indication, went through a phase 1/2 trial and established essentially a single administration durable treatment for a condition called cystinosis. And that program is now being led by Novartis through a new trial in pediatric patients. And so what we've refocused the company on is our preclinical portfolio of assets, the lead program of which is called PPL-001, which is intended as a one-time disease-modifying treatment for Friedreich ataxia. And we feel that with the partners that we have and a really solid disease foundation, that we have a great path to the clinic, along with CIRM funding to back the program. So the team is small but mighty. The real star of the show is Dr. Stephanie Shirky, who's the scientific founder of the company. She led the cystinosis program from inception, bench to bedside, through the phase 1/2 trial with the idea that you could gene modify the patient's hematopoietic stem cells and reintroduce them for long-term durable treatment in which the monocytes in the patient deliver the protein that's missing. But there are other experts on the team: Chris Mason, Jeff Ostrove, Eric Adler, and Farah Sheikh were co-founders along with Stephanie. And then there's a series, obviously, of kind of platform partners that we're working with to advance the therapy. Manufacturing, for example, at Thermo Fisher's facility, off-target analysis with the Maxcyte group and Chromatid, and then backing from CIRM and support from the Disease Foundation as well. So this is just a summary of our therapeutic pipeline. As mentioned, the first program in cystinosis was taken through a phase 1/2 trial that reported out a few years ago, and that has been picked up by Novartis and now being advanced in a new clinical trial in which they're expanding the label to pediatric patients. Our company is very focused right now on taking our lead asset, PPL-001 for Friedreich ataxia, through IND, slated for the end of next year. But we do have additional programs in development that are funded by CIRM and other sources, one for Danon disease, and then another one that's early stage discovery in Alzheimer's. And CIRM has provided a lot of funding to this modality and this program. We have been selected for potential additional funding from them, and the programs have received RPD and orphan designation. So the nature of these genetic diseases is often one in which there are multiple tissue systems that are damaged, and so that's really the challenge that this approach tries to solve. It's kind of front-loading the technical risk of doing the ex vivo autologous manufacture of the product with this long-term potential benefit to the patient, where they might see multiple disease-affected tissues improve function. And so that's really the thesis of the company. We think it provides an advantage in leveraging the body's natural system for delivering protein to the body. And the schematic on the right is just illustrating, for Friedreich ataxia, for example, you get an effect of the disease pathology across multiple tissues just from the loss of function or loss of expression of one protein. And so an interesting thing about the hematopoietic system and the myeloid cells is that they engraft durably in the bone marrow, and then they shed monocytes, which then go to different tissues in the body and engraft there. And they have a property where they transfer cytoplasm from the donor cell, gene-modified donor cell, for example, into the other cells in the tissue. And it's not a mechanism that's really deeply understood, but it's been observed in vivo, and in this case, just shown here in an in vitro assay. So that's the idea, is to gene-modify the patient's myeloid compartment and allow that myeloid compartment to deliver the protein payload broadly through the CNS, the heart, other affected tissues of the body. The validation of this through the phase 1/2 trial in cystinosis had a number of different data points, obviously, but one of them was to look at the reduction in cysteine levels in circulating leukocytes. And so in this condition, you have a buildup of cysteine levels that's uncontrolled and damages all the tissues effectively of the body, some more than others. And so really one of the ways to monitor the effect of the treatment is to look at the level of cysteine in the patients over time in circulating leukocytes. They certainly looked at other tissues as well, but that's some of the data shown here. And so now this program, having been licensed to Novartis, is really for them to develop, and we're very supportive of that, but it's really their program to speak about in future meetings. What we are very focused on is this program that we have for Friedreich ataxia, which is a very devastating condition in which you have a series of dysfunctions and pathologies across multiple tissues, notably in the CNS, where patients' degradation of the CNS results in many of them being in wheelchairs fairly early in life. And the morbidity of the condition, it really comes to bear when there's cardiomyopathy and other pathologies of the heart that can be life-threatening. And so it's sort of a multi-system disease that there are some treatments that have been approved. Skyclarys is the first one, other treatments in development, but we think we're offering the only treatment in development that might be able to target a whole series of tissues and potentially halt progression or maybe even reverse it. And so the product, PPL-001, is a product in which we harvest the patient's hematopoietic cells, we gene correct them, which is to say we remove the pathogenic intron repeat expansion that causes misregulation of the frataxin gene. And this is done with a gene correction approach with 2 guide RNAs. And so really one of the most important ways to de-risk the program was to just make sure that the guides that we're using are not having untoward off-target editing, and so we've done that with Maxcyte and Secure and some other groups, and that's looking very positive relative to other commercial programs. So we're very happy about the precision of this excision treatment of the HSPCs. And we think that this approach avoids the risk of overexpression of frataxin, which has been shown in certain settings to be toxic. And so there's sort of an advantage in just correcting the deficit that's present in the vast majority of the patients. So the disease model that we're showing data for here is one in which it mirrors the patient journey, where the animal in this case is an animal that has the human frataxin gene, and it's knocked out for the mouse equivalent frataxin gene. And the human frataxin gene that's been knocked in has a large number of the repeats that are part of the pathology of the disease. And so we can use our ex vivo gene excision tool, our RNP, that we're using in the human cells to test the effect of this in vivo in this particular animal model. And one of the results of that is you see a significant increase in frataxin expression, which is the goal of these therapies, is to increase the level of frataxin to as close as possible to normal levels across multiple tissues. In this case, it's showing total expression in heart in the middle, in the cerebellum on the left, and then in terms of mitochondrial associated frataxin, that's shown in the data on the right. And relative to wild type, followed by untreated versus mock treated, and then PPL-001 equivalent treated, there is a significant improvement in the expression of frataxin across multiple tissues. So then the question of function, it is a little difficult to establish functional readouts in the animals because they're not as affected by the condition as what is observed in human. But nonetheless, you can see improvements in motor function, gait and balance is shown on the left. You can see increases in heart mitochondria decreases. And then interestingly, one of the symptoms that Friedreich ataxia patients have is they develop a diabetic phenotype. Not all of them, but a significant proportion of them do. And if you look at the islets and the amount of insulin production happening in the islets, you do see a rescue of that as well in the animals. And so it does seem that the treatment in the preclinical model is having this multi-system effect. So high level, and I'll probably give you 5 minutes back, but really we are a preclinical stage company that has a clinical proof of concept data set for a program that's licensed to Novartis, and we're pursuing the 001 program through IND expected at the end of next year. We have been selected for prior funding from CIRM. We've secured, a variety of different regulatory milestones with FDA, established precise on-target editing for our product, and now we are in the midst of completing our engineering runs for IND-enabling studies. So really the next step for the company is to raise our Series A and pursue the program into the clinic. Thanks for your time.